Miss Lauren. Let me see what she couldn't say. And she is talking about our antibiotics clinics.
Alright. Hello, my name is Lauren Benzel. I'm a sophomore at St. Petersburg College, and I'm here to ask the question, are antibiotics killing us? And in some cases, yes. Not from the antibiotic itself, but rather the repetitive use of antibiotics. Because unfortunately, the more and more we use these antibiotics, the more likely we are to create superbugs. Superbugs are bacteria that have become resistant to antibiotics. And these are very dangerous bacteria, such as MRSA, which is a strain of Staphylococcus aureus, Streptococcus pneumoniae, E. coli, and Cinebacter, on top of many, many others. And unfortunately, the solution is not to increase our antibiotics, because if we do that, natural selection is only going to take its course, and the bacteria are only also going to evolve and become stronger. So what do we do about this, and is there a natural solution? And I say yes. So I decided to test essential oils and see if these would actually kill the bacteria and in turn slow down the adaptation rate. And an essential oil is an oil extracted from a plant. So pretty much if there's a plant, there's an oil to go along with it. There's hundreds and hundreds of these, and they're used in perfumes, food, things like that. But most commonly you'll see them in aromatherapy. And people claim that these have the antimicrobial properties, so I decided to see if that was true. So how did I do this? The first step was to culture the bacteria, and I used 5 of the 6 escape bacteria, which are the bacteria commonly used to test antibiotics. So the 5 I cultured were Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Pseudomonas aeruginosa, and Enterobacter aeruginus. So after those were cultured, we made the nutrient agar, and we had 7 flasks. Each had 1.6 grams of the powdered agar in it, but had different concentrations of water. The first one had 100 milliliters, second 99.9, 99.5, 99, 98, 95, and 90 milliliters of water, and the amount of water would then determine how much oil was added later. So the flasks were then autoclaved to sterilize it, and it was time to then mess with the bacteria some more. So 3 milliliters of each bacteria was added to 1.5 milliliters centrifuge tubes, having 1 milliliter in each tube. And those tubes were then put in the centrifuge and spun at 10 RPMs for 2 minutes. And what this gave us was a small pellet of the bacteria at the bottom, which allowed us to pull out the agar which the bacteria was growing in and resuspend it in PBS. So then after that, the essential oil was added to the agar. And as previously mentioned, the amount of water added to the powdered agar determined how much oil was added. For example, the flask that had 90 milliliters of water had 10 milliliters of oil, and the flask that had like 99.9 milliliters of water only had 0.1 milliliters of oil. So then it was time to put it all together. And it was all put in a microtiter plate, which is this right here. And the first row right here would be our negative control, which was bacteria with just PBS, which is essentially just saline. The positive control right here is the bacteria with the agar that had no essential oil in it at all. And then from there, it just ascended upward with the amount of oil that was added. Each bacteria was tested in triplicates, so each one got its own 3 rows. So 100 microliters of the oil agar solution was added to each well, with the exception of the negative control. In that case, 100 microliters of the PBS was added. And then 100 microliters of the respective bacteria were added to the wells. And after that, it was time to read the plate after everything had incubated and get our results. So the first oil that was tested was eucalyptus oil. And this spike right here was just the positive control, because there was nothing to combat the bacteria at all. And then down here is just where there was PBS and not a lot grew to begin with. And as you can see, for Klebsiella pneumoniae and Pseudomonas aeruginosa, there was definitely a downward trend, especially with Pseudomonas aeruginosa. There was a huge drop here. With Klebsiella pneumoniae, still a pretty big drop. It just went kind of more gradual descent. Staphylococcus aureus, eucalyptus did not seem to have a huge effect at all, if any. And Klebsiella pneumoniae still had some spikes here and there, but for the most part, it's still lower than the control group, which is what we want. So the next oil tested was rose oil. And rose oil, for this one, we tested Klebsiella pneumoniae, Staphylococcus aureus, Pseudomonas aeruginosa, and then we also added Endococcus faecium. And Endococcus faecium, it seemed to work pretty well, but what's interesting is that it actually starts to go upward with the more oil that was added. But then down here, with only 0.5% oil, it seems to have dropped. Klebsiella pneumoniae still seemed to have a pretty downward trend. Staphylococcus aureus, again, didn't really seem affected by the rose oil. And Pseudomonas aeruginosa is kind of going all over the place here. So then the third oil we've tested so far is oregano, and oregano worked incredibly well, a lot better than the other two. So Endococcus faecium wasn't really affected. Staphylococcus aureus was kind of affected, but what was really notable was the Enterobacter and the Enterobacter, excuse me. And the Enterobacter hardly had any growth at all with the oil, which is really significant. And it's what I hypothesize is that it's the gram-negative bacteria, as opposed to these two, which are gram-positive bacteria. But we're still not done, because then it was time to test for biofilm. And biofilm is like a sticky bacteria, which is a lot harder to get rid of because it'll kind of weave its way and kind of hide itself, so it's a lot harder to get rid of. So to do this, we washed out the plate to get rid of any planktonic bacteria, and then the wells were stained with 125 microliters of 0.1% crystal violet, which would stain the bacteria and then allow us to see it better. And after that, after letting it sit for 10 minutes, the excess crystal violet was washed out. It was let to air dry for a week, and then alcohol was added to the wells, and it was time to read the plate again. So for eucalyptus oil, it seemed to have the pretty much the same kind of trend going on. With Staphylococcus aureus not really affected, Klebsiella pneumoniae kind of downward, and Pseudomonas aeruginosa, it tended to work best on. Rose oil seemed to, again, have a pretty similar trend. With the Endococcus faecium still working pretty well, although it seemed to work better on the planktonic bacteria than it did the biofilm. And then Staphylococcus aureus just doing its own thing here. And Klebsiella pneumoniae, though, it seemed to have a more uniform downward trend with the biofilm than it did with the planktonic bacteria. And then, unfortunately, oregano, still in the process of getting the results for that one, but I do have high hopes considering the results for the planktonic bacteria that I saw. So in conclusion, essential oils do have an effect on killing the bacteria. Granted, some work better than others, but as you can see, like for oregano with the Endococcus faecium, it had a huge effect on the bacteria. And so it's definitely something that should be looked into. For the future, I am going to continue to test oils on the same bacteria I've been growing. However, I want to also test the effectiveness of this process on bacteria like P. acnes, which causes acne, and S. mutans, which cause cavities. So I want to thank St. Petersburg College for allowing me to use their lab to conduct my research. I also want to thank my mentor, Dr. Shannon Ulrich, who helped me through this process, and my lab partner, Michaela Sapp, who made all that pipetting a lot more bearable. Photo credits, and are there any questions?
Have you looked at how effective it is versus using the antibiotics, like done a comparison of?
I haven't done it as a comparison with the antibiotics because right now I just want to see if they'll like kill it and see if it's even an alternative solution at all.
Okay.
Yes.
I just have comments. I remember listening to one of my teachers talk to me about this during my freshman year.
Yeah.
And after she told me this, it really, whenever I get sick or anything, I don't take antibiotics. I usually just take it and wait for it to go away.
Yeah.
Because I know it's just contrary to making it stronger.
Right.
And just seeing it's really eye-opening.
Yeah.
Thank you.
Yes.
Has your research applied to like other drugs aside from antibiotics, like immunosuppressants or?
No, I haven't really been looking so much at like the drugs that are causing the bacteria to become antibiotic resistant. I'm just seeing if the bacteria that are becoming antibiotic resistant, if there's another way to kill them besides the antibiotics or other kinds of drugs.